Hello, my name is Milan Doldan. I am in Professor Rogers' microbiology online lab class. The topic of my midterm video today is going to be about peer culture. I'm going to go over what a peer culture is, what procedure we use to make a street plate, what outcome are we looking for when we make this street plate. I'm going to explain, why it's important to make this street plate from a clinical specimen or make a peer culture from a clinical specimen. And lastly, I'm also going to talk a little bit about, what we might do to help treat a patient that might have this type of infection from this bacteria. So pretty much in microbiology, a pure culture is just a single type of organism or a single species of an organism. A microbiology culture is just, the method of multiplying microbial organisms by letting them reproduce in a controlled laboratory condition. And a street plate, or the street plate method is just the procedure or technique that we use to help isolate these different colonies from contaminants. And, what we're looking for is, pure isolated colonies. So pretty much this procedure starts by you sterilize a wire loop in a Bunsen burner, a flame from a Bunsen burner. You let it cool a little bit, and then once it is cooled, by sitting for a few seconds, you dip it into a sample that contains a mixture of this bacteria. Now, when you pick this loop out of the bacteria, you may pick up hundreds, thousands, sometimes millions, and, of these bacteria. So pretty much what you're gonna do, you're just gonna spread it across the surface of a plate, And I drew a picture. There's a few ways you could do it. There's a few different patterns, street patterns that you could do. The first one that if you just have one bacteria, you're just gonna do back and forth, downwards motion. And then another pattern that you could do is back and forth, downwards. Or, you may do it in the corner, you may do it on half, but you're always gonna intersect them. So then after that, the cells are going to start to divide, going to start to form colonies. Oh, well, actually, You're going to put it into an incubator. Then it's going to start to divide and form colonies. And then what you're going to see all these little single cells forming these colonies, and they're going to be genetically identical clones. And in this isolated colony, it's going to grow bigger. And the reason why it's growing bigger is because there's less competition for these nutrients. And, when you do a street plate, the outcome that you're looking for are those isolated pure colonies, or even just one isolated pure colony. And the reason why we want them to be isolated colonies is because we can then study and identify them. We can look at the characteristics, see what's different, see what's similar about this pure culture. And also, the isolated colony, it's going to have genetically identical bacterial cells that are coming from the mother cell. They stem down from this mother cell. And the reason why we want to make sure a pure culture, is, why we take it from a clinical specimen is because we need to see where this bacteria first started. We need to see why it's happening. Maybe if there's a disease outbreak, we need to see what caused it, where is it coming from, and we got to stop it. Now, it's also important for proper disease diagnosis. So, if you're a patient, you go in, you're sick, you need a treatment plan. So in order for this treatment plan to start, you got to see where this disease is coming from. It's going to be a mystery if you don't know where it's coming from, what started it, what kick-started it. And, if you have that treatment plan, you can, further treat other patients that have these microbial infections. So in order to help a patient that may end up with this type of infection, you're going to need antibiotics. You're going to get antibiotic prescribed from your doctor. They're going to assess your symptoms, your signs, what you feel, all that. Then the antibiotic is just going to pretty much help kill off the bacteria or either prevent it from further multiplying. And what also it does, it helps the body's immune system. It's going to fight off that bacteria. And the type of antibiotic will all just depend on what your doctor suggests is right for you, what you can handle, what caused this infection, what type of bacteria is it that you're dealing with. And, it's all going to just depend on, can you have this antibiotic, can you not? All those types of things play a role in it. And that is the end of my video of pure culture. I hope you enjoyed, and thank you so much for watching. Bye-bye.